Yes for giving me this opportunity. Today I am going to discuss my research for this prestigious award in front of Talwar sir and all the my senior and teachers. So the nature of maternal newborn immunity and myth of hepatotourism of hepatitis B in mother to baby vertical transmission. So before going to detail in my research, just I have one background, one slide. The hepatitis B is a DNA virus which is non-cytopathic to the cells, and it can lead to the liver cancer and the cirrhosis in the severe cases. So the hepatitis B infection in India and worldwide approximately 400 chronic carrier, and India has approximately 45 million. That means 3 to 4% among the 1.5 billion population. And so the chronicity is depend upon the age, and it's inversely proportional to age. If mother is infected with hepatitis B, the chronicity will be high compared to the adult, which is only 5 to 10%. So in India, the prevalence of pregnant female is 1.2 to 1.6%. This is study from the Delhi and the Bhopal. So what are the challenges we have? First, if hepatitis B infected mother, one mother transmit the virus to their newborn, but another mother is not transmitting. Why? Is maternal immunity playing any role, or the newborn immunity playing any role, and what is the role of placenta? So I am just going to brief with this. So what we have done, we have screened a pregnant female approximately 45,868 from Delhi, 18,461, and from Bhopal 27,407, and we found the average prevalence was 1.63%. We follow these positive mother till the delivery and further divided into groups, transmitting group and non-transmitting group on the basis of transmission in the newborn at birth, and we follow the healthy controlled way for the comparison. So what we have done, we have taken placenta, we have done entry receptor expression studies in placenta and the viral markers, which their infection in placenta or not. We have taken peripheral blood, and we have from blood we have taken plasma, PMC, RNA. We have done the serological profile, cytokine analysis, and immunophenotyping, and HBV specific responses and mother baby pairs, and we have done the RNA-seq analysis also. We also done CBMC, cord blood mononuclear cell. So we know that how the immune response work. If dendritic cell recognize the antigen, they present T cell, then T cell. By the CD8, they have cell-mediated immunity, they secrete pro-inflammatory responses, and the other aspect that humoral immunity by CD4 and TFH cell help B cell, and they secrete antibody. So we have the protections. What happened in this mother-baby pairs? So what we have done, first we found that increased dendritic cell frequency in HBV specific response in mother who is non-transmitting to their newborn. So this is the green color for the baby, and this is red color for the mother. So you can see the transmitting mother have less MDCs and their newborns also, and the same happen the PDCs. We also look the HBV specific responses by looking the IL-12 and the IL-10. So we know the IL-12 is required for the knife cell T cell differentiation, and we found this MDCs have defective in IL-12. They are not able to secrete IL-12. But if you see the PDCs, they are secreting more IL-10. That means they have immunotolerant nature. We have done other work, but I'm not showing here. We also look for the TLR signal pathway which is required for the hepatitis B recognition. That was also impaired. So now we want to see what happened in the T cell mediated HBV specific responses. We found that CD4 specific secretion against HBV, which interferon gamma down, IL-17 decreased by the mother and their newborns. Similar, the CD8 will happen. We have limited sample because this is the at birth study. We have very less sample at the birth. So we look for the anti-inflammatory response in mother only. And we found that IL-10 and TGF-beta, HBV specific by CD4 and CD8 will also increase in the mother and their newborns pair. We also look for the B cell, and we have categorized three: total B cells by CD19 positive, and mature by CD19-10, and the plasma B cells. And we found that mother who is transmitting and their newborn have high and mature B cells, and they have less plasma B cells, which required for antibody secretion. And what will happen, this immature B cell directly proportional to HBsAg, which is an infectivity marker. So if you have high immature B cells, you will have high infection. If you have high plasma B cells, you will have less infection. So this is inversely proportional to a plasma B cells number. Then you look why this ammaturation happened. Videsh, Dr. Nimesh Gupta had discussed about the TFS cell. They help B cell in the plasma B cell formation. And we found the defective TFS cell in mother and their newborn. We also look for ICOS expression in these cells. This is also defective. So, and we did RNA-seq, and we looking for the genes. What happened to the specific genes in the B cells, CD8 and CD4 specific. And we found this non-transmitting methylome high signal cytokine which required for the antiviral immunity by these cells also. So we look for the TLR pathways. So these are the down regulation by the transmitting mother. So they have most of the TLR signage impaired in that. We have looked for the T cell response. Most of the genes are impaired by the gene. So now what we concluded is mother who is transmitting the virus, they have less TLR signaling, impaired dendritic cell presentation because of they have defective naive T cell differentiation in the TFH, HBV specific CD4 defect, defective HBV specific CD8 T cell, defective HBV specific TFH cells, and they leading to the impairment of inadequate and neutralization and impaired cellular immunity. They leading to the enhanced HBV vertical transmission. We want to see what happened to placenta. So till now, no one studied the receptor expression. Because why this hepatitis is a hepatotropic virus? Because it's a receptor expressed on the liver only. We first time shows that this receptor is acyloglycoprotein receptor, ASGPR, a transmitting mother have higher expression compared to the non-transmitting and healthy mother. So, and we also look for another receptor, sodium tarcholorate co-receptor protein. This is a receptor which help in the entry of hepatitis B. And we found the first time we have showed recently that this is also expressed in the placental cell. We also look for the HBE antigen. This is a marker of hepatitis B replication. And we found this in the placenta. They have it. We look the mother who are HBE antigen negative in circulation, but they are positive HBE antigen positivity in the placenta. So this showed that this is a another home for the virus. And we also look for the cccDNA, this covalently DNA, which is considered the mini chromosome hepatitis B. This also found in the placental sample. So what the summary is this, placenta is another home for the HBV virus other than the liver, and the presence of HBV interceptor, SGPR and SGP on placenta along with cccDNA and replication marker suggests that possible role of this organ in the vertical transmission. Targeting this mechanism may eliminate the reduction. So mother, baby and placenta, the unholy trinity of hepatitis B vertical transmission. So this is the publication we have so far, and this is the publication recently from my lab related to the vertical transmission. And so this is my student Garima Gar, Kajal Patel, Priyal, Vijna, Meenu, Dipesh. So they are working in hepatitis B vertical transmission. My supervisor, Professor Nirupma Patish, Professor Kumar Sharir, and Dr. Anand Singh from AIIMS Bhopal. And these are the collaborators. After my PhD, I am working with them. And the Department of Microbiology, I have funding from DST Inspire faculty, serve core grant, and I have association with Nationwide Children's Hospital, USA, and University of Maryland, USA. Okay, sorry. Thank you so much.
So, questions from judges? Dr. Pradeep?
Dr. Ashish, a very nice presentation. I'm just wondering whether you have checked that the difference in the transmission from mother to placenta. Did you check the any polymorphism of the receptor that is responsible for viral uptake?
Yes.
And another thing is that what I want, because you have shown that virus is entry is mainly because of the dampening of the immune response. So what might be the receptor through which the virus is mediating this immunoregulatory effect, and whether it is happening in the placenta interface like that?
Yeah. So, sir, the first question, we look for the 21 genes which are responsible early recognition, and we found the mutation in NLRP3 gene and ISG15. So this data is presented as abstract by Garima, my student here only in this conference. And the second about the vertical transmission happened by 3 routes. It's intrauterine, perinatal or postnatal. We believe it's happened before delivery by transmission, before delivery. Why we are showing, there are studies in hepatology journal which shows that MDSC, matrix stem cell, they express ASGPR and infection happened. And they have done in, before delivery the fetus because of abortion. And they study the HBV infection in the fetus so before delivery also. So we believe that. And forgot the question.
Another thing is that polymorphism I was talking about.
Yes, so we found there is RSID, I don't remember, my student presented a study only. We found NLRP3 mutations, SNP, NLRP3 genes, NIS ISG 15. But recently we have done by Agenema study technology in that.
So does it correlate with this
Actually,
particular?
sir, we have like very limited sample size for the SNP because we have given 20 sample and we found one, so this percentage we not able to correlate it because the vertical transmission is not that much. We have only 17%. So if you follow 100 mother, actually the definition of vertical transmission is the positivity of baby after 6 months. Because when you deliver, government have a scheme for the hepatitis B vaccination and immunoglobulin. So that time virus may be neutralized. But if you have like cccDNA or the DNA integrated with the genome, what will happen after 6 months again will. So we are telling the vertical transmission after 6 months of baby. So this is a study started 2 years and we have around 10 the following the vertical transmission is confirmed.
So I just have two questions here. So one is whether you looked for the presence of type 1 interferons because it's a viral infection. I didn't see any documentation on that.
Yeah, I have seen them.
The type 1 interferons.
Actually, I have seen interferon gamma. I didn't see interferon. Yeah.
Because in most viral infections, the main antiviral agent is the interferon alpha.